Hi everyone, this is your instructor Kwame. This lecture is the final part of our lecture on microbial growth and control. Specifically, this component will focus on microbial control. For objectives, we'll look at some terminologies, the meaning of death rate, microbial considerations when we are controlling microorganisms, the methods and the modes of actions or the methods that we use. And so we'll look at the physical methods of microbial control and chemical methods of microbial control. I have this cool cartoon here where you have these healthcare professionals who are talking about patients, that they whine about MRSA, and then they moan about the disinfectants that are used in the hospitals and that there is no way of pleasing them. And that is the relationship that we have with most of the methods of microbial control, especially when it comes to settings like the hospitals and institutions of learning. So for terminologies, sterilization is a very important term when it comes to microbial control. And sterilization means the complete destruction of all microbial lives, including spores. However, there is a caveat to this definition, and the caveat is that some disease-causing particles like prions are not destroyed in sterilization. You would have to modify the method of sterilization to achieve destruction for particles such as prions. Another exception is that for commercial sterilization, the term sterilization is used quite loosely, in that only the microorganisms that cause spoilage and are pathogenic are the ones that are destroyed. Not all microorganisms are destroyed in commercial sterilizations, and that's an important point to note as well. So, for instance, canned food are treated at temperatures that will get rid of Clostridium botulinum, which may cause botulism, but not necessarily all the spores that exist in the canned food. Then there's disinfection. That's control that is directed at destroying the vegetative cells, only the harmful cells, especially of pathogens. And so we use chemicals and we use physical methods as well. And then there is antisepsis. So antisepsis is disinfection of living tissue. So if disinfection is done on a living tissue, that's antiseptic. So if you go to the hospital and you're about to be given an injection, that area is cleaned so that it's sterile, and that's an example of an antisepsis. That is not disinfection. It comes down to what surface it's being used against. Then there's degerming or degermination. That's the physical removal of microorganisms, and that is a process that we see in the use of soap to wash our hands. That most of the process of removing these microorganisms is by degerming or physical removal. Then there is sanitation. That's more of a public health term when you lower the number of disease-causing organisms in the environment and to reduce the spread of infections. It's the same procedure that is used in dishwashers and glassware in bars. So you have very high temperature washing in terms of restaurant glassware and china and tableware, or they wash these glassware in a sink and dip them in a chemical disinfectant. So those are some of the methods that are used. Still with terminology, you have certain suffixes that gives you an idea what a particular chemical does. So we have bacteriostatic, and those will stop the growth of bacteria. And once you take them away, the bacteria will start growing again. So the static tells you that there's a stasis, there is a stoppage, there is a pause. And so if you look at bacterial growth and time, it's going exponentially now. You add an antimicrobial. What kind of antimicrobial is it? If it is a bacteriostatic, the growth will remain the same like that, and then it's just going to continue being at that level. However, if it is a bactericidal, which causes the death of the bacteria, then what's going to happen is that there's going to be a sharp decline in the number of cells of bacteria that are in that particular system. So this table summarizes what I just went over with you. So there is sterilization, destruction, and removal of all forms of life, including endospores. And this is usually done by steam under high pressure. You can use an autoclave to do that. And there's commercial sterilization, where the most important in terms of disease-causing organisms are the ones that are targeted. So you have endospores for Clostridium botulinum that are targeted in such an instance. Then there is disinfection, there is antisepsis, there is degerming, and there is sanitization. So if we look at the rates of microbial death, since we are talking about microbial control, it is calculated as the number of death under certain standard conditions. So we look at death per minute and the rate at which the organisms are dying. So example, 90% per minute of dead organisms. Okay. If we look at the practical graph, if we look at, so you have, say, log 6 of cells, in terms of you have about 1 million cells, and you are applying a particular bactericidal, a chemical that was going to kill the bacteria, and you look at a practical graph in terms of the death rate. That's how it looks like, the blue line. What you will notice is that by the time it gets to 3 minutes, you don't actually see any difference in the number of bacteria that dies. But that is not the reality, because you still have bacteria and you're still dying since you're still applying the bactericidal. So for commercial purposes, we rather have this graph, which is a log graph that tells us, so each box represents 1 log, that per every log, which is the time that we're looking at, so in this case per every minute, okay, you have 90% of the population of the bacteria that exists at that time that will die. So that's a more realistic way of looking at the number of organisms, and this is what is used in industry to guide the standards in terms of quality that are maintained in the industry. There are certain factors that will influence the number of bacteria that die if particular agents apply to them. And some of these factors are the type of media that they are in. If they're on surfaces, just plain surfaces, it's very easy to deal with them. But if they exist in, for instance, in biological samples, then it means they exist in the midst of some fluids and some certain tissues, so it's more difficult to deal with them. Then there's temperature. Is it high or low temperature? The pH as well. So these environmental factors are important factors that determine or influence the death rate of microorganisms. In addition to these environmental factors, you have time of exposure and you have microbial characteristic. In terms of the number of microbes, which is also a factor. So for instance, if you have low population and you apply a constant bactericidal to these microorganisms, for the low population, you're going to see that the load decreases and eventually the number that survives becomes zero very quickly. So that at time 3, there are no bacteria existing. However, if the load is greater at time 6, that is the time that you might not see any bacteria. In terms of microbial characteristic, you have different bacteria. You have gram-positive, you have gram-negative. You have those that have resistance because of their cell wall, such as Mycobacterium, which have a waxy outer covering made up of mycolic acids. If you look at Pseudomonas has these porins that help regulate what can get into the cell and what cannot, and so prevents bactericides and antibiotics from getting into these cells and to destroy the bacteria. Gram-positive, gram-negative. Gram-positive is more difficult to deal with. Gram-negative rather is more difficult to deal with because they have the extra outer membrane. Gram-positive, it's easy to deal with because you have this huge composition of peptidoglycan as part of its cell wall and cell membrane, and so these differences influences how they are treated. So that's an important component in terms of control methods, that the bacteria and how it behaves influences what we use to treat them. So Mycobacterium, which causes tuberculosis or leprosy, it has this waxy lipids in its outer covering, and that makes it difficult for most treatment of control to actually be effective on them. Now, if you want to control microorganisms, these are the things that you want to look at. You want a method that is inexpensive, that will act quickly, that wouldn't destabilize the system, and that is safe. And in terms of the factors that we'll look at what site are we going to use. Is it going to be a local site? Is it going to be some kind of systemic thing? And then you look at susceptibility. How susceptible are the pathogens to the agents that you want to use? And then environmental conditions such as temperature, pH, and pressure. For the mode of action, you want agents that will destroy the protein, denature the proteins of the microorganisms, destroy their nucleic acid, or damage their lipids or proteins in the cell membrane. So if you tamper with their enzymes, you tamper with their genetic material, nucleic acid, or you tamper with the cell membrane, you would be successful at destroying the bacteria. So in terms of the methods of control, you have both physical and chemical methods of control. Physical methods would include temperature, desiccation or drying, filtration, using osmotic pressure, and radiation. For temperature, we have moist heat and dry heat. For moist heat, you have boiling, autoclaving, pasteurization. Dry heat, you have direct flaming, incineration, and hot air sterilization. Now, when we look at temperature vis-a-vis controlling bacteria, there are 3 important indicators. And the first one is the thermal dead point. So you want to know the minimum temperature for complete microbial death in 10 minutes. So within 10 minutes, what's the least temperature at which you can destroy that particular microorganism? Then there is thermal death time. That is the minimal time for complete microbial death at a specific temperature. So let's say you pick a temperature of 40 degrees. How long does it take for these organisms, when exposed to a 40-degree temperature over a period of time? Then you have decimal reduction time. So that's the time in minutes at which 90% of the bacteria is killed at a given temperature. So at a particular temperature, say 90 degrees or 100 degrees, if that's the time that you have 90% of the bacteria killed at that temperature, then that is the decimal reduction time for the bacteria at 100 degrees. So for moist heats, boiling is used to kill vegetative cells, so it doesn't kill spores, it doesn't kill viruses, but it's able to kill bacteria that is not sporulating, fungi, protozoans, and some viruses as well, but most viruses no. And so this is routinely recommended for housewares and cooking utensils and stuff like that. Then you have autoclaving, which is a pressure system. So you have 15 pounds per square inch in terms of the pressure. And then you have a temperature of 121 degrees. And this is run for 20 to 30 minutes. So it kills all microorganisms or disease-causing organisms except prions. And then it's able to sterilize. We use it to sterilize media and instruments and solutions. And it's heavily used in microbiological labs. The autoclave, very indispensable. And in autoclaving, there is a tape that is used, the adhesive tape that is used in autoclaving. Now, it has certain features. This is example of the adhesive tape that is used in autoclaving. It has a number of features that helps us to actually identify what's going on when we're autoclaving. So under high pressure, we have to know when we have reached the right temperature. And what the tape does is that, the right temperature in terms of how long we have autoclaved the content. The autoclave tape works by changing color after it's exposed to temperatures that are commonly used in sterilization. So typically at 121 degrees Celsius in a steam autoclave, you're going to see a change in this tape, in the color, and that will indicate to you that it's been in there for enough period of time for the contents to be sterilized. Then there's pasteurization. Remember Louis Pasteur? So exposing the content to temperature or heat that is hot enough just to kill the foodborne pathogens without destroying the food or changing the taste of the food. And this is still used in the dairy industry to preserve milk for consumption. Then we use the ovens and incineration, for these are dry heats. When used with water, then it becomes moist. So without water, we have a dry heat option. It's not as efficient as moist heat, however, and it takes a longer period of time. So note some of these differences that I'm mentioning. And then for incineration, you have to flame or incinerate, and If you were to do a live lab, that is what you would do with your Bunsen burner. That when you pass it through the flame using the tip of the inner cone, that is an example of incineration. You could also use hot air sterilization. That would be dry heat. Then for cold, so aside heat, you can use cold to preserve food as well. And that would depend on the number of microorganisms and how intense the physical method in terms of the code that we're using is. So if we use refrigeration, you're talking about 0 to 7 degrees, it's going to stop the growth of the microorganism, but it's not going to kill the microorganism. So it's bacteriostatic. An examples of organisms like the psychophilics are able to survive at this temperature, and that over a longer period of time, they will eventually cause food spoilage. Then there is freezing, that's below 0 degrees. It slows metabolism, and the interesting thing about freezing is that it's not actually the freezing that kills the bacteria per se. It is actually the thawing that causes the most damage to the bacteria. That's pretty interesting. That as it thaws, because it thaws way quicker than the rate at which it freezes, it kind of throws the bacteria off in terms of their temperature and all that, and the whole process ends up destroying them. So filtration also exists. That's another way of controlling microorganisms by passing the liquid through a membrane that has smaller pores, which will prevent the microorganism from going through. And when it's vacuum assisted, it actually improves the process. An example of a filter that can be used is filters that are made of nitrocellulose, because they have very tiny pores in them. There are certain things that we cannot sterilize by heat, and these are some of the things that we use filtration to sterilize, like vitamins and antibiotics and enzymes that heat would destroy. You have high efficiency particulate air filters, which are used in labs and in the hospitals as well to filter out air. So it creates this aeration system that moves contaminated air out of the way, creates a sterile environment in the cabinet, so that whoever is researching or doing experiments there can do experiments and carry out tests, and we can trust the results that it is not a doubtful results, because the environment was sterile. And so it creates what we call a laminar flow cabinet in the laboratory. Then there is desiccation. This is something that has been with us since the beginning of civilization, that when you dry food items, it stays longer. This is also an example of a bacteriostatic. So immediately you introduce moisture, the bacteria or the microorganisms are going to grow again. There are certain organisms that are resistant to desiccation, the removal of water, and Mycobacterium tuberculosis is one of them. It can stay without water for months. And note also that in addition to these organisms, you also have organisms that are spores, are able to form spores. You have others that are viruses, and these are all resistant to desiccation. Osmotic pressure kind of operates in a similar way, but it uses the concept of hypotonic and hypertonic solutions, or osmosis. So you have high concentration of salt or sugar, and then you have a hypertonic environment. So water is going to leave the microbial cells into the environment. So all these charm and very salty preparations that we have, it's not like the bacteria don't have access to them, but when they enter these food preparations, they end up dying because of the system that is set up there. So it's used for food preparation, but you still may have microbes that will enter, especially fungi. So they are not antifungal. There's radiation, where different wavelengths of light are used, gamma rays, X-rays, and UV light. So if you use ionizing radiation, which is widely used in the food industry and the pharmaceutical industries, they tend to be very highly efficient. The non-ionizing ultraviolet radiation, they are used to damage DNA and kill they're able to damage DNA, both microbes and eukaryotes, but they kill organisms in the air. There's a direct exposure, but it's not very safe for us as humans. So this food has been preserved using ionizing radiation. This is regular food that has not been preserved, and you can see that it takes a very short period of time. And we have experienced it ourselves, where you pick food item and natural stuff that hasn't been ionized, and it's not going to last for too long before it goes bad. And then you have UV lights, which are used in laminar flows as well to sterilize the environment. Radiation in microwaves are commonly used as well. When you microwave a food, you are applying direct radiation to the microorganisms that may be present in the food. So it kills the vegetative pathogens, but if they are spores, it wouldn't be able to kill them. If they are prions, they would be able to kill them. For solid foods, they don't heat evenly. So if they are contaminated with trachinoloses, pork worms, then most likely the person who consumes them may also suffer from the infection. Then we have chemical methods. You have living tissues and inanimate tissues, or objects that are applied to, that these chemicals can be applied to. The idea is to reduce the population of these microorganisms to levels that are safe, and it's mainly remove the vegetative forms. It's more of a public health thing. So the idea is not to create a sterile environment. It is just to reduce the amount of organisms that are there, so that even if people end up picking these microbes, the concentration is low enough and our immune system will be able to fight them. So if whatever disinfectant you end up using has to depend on what the content is and what surface you want to use it on. Okay, so you have to read the label and make a decision on that. The nature of the material is important as well. Is it organic? Is it artificial? So you take all that into consideration. How then do we evaluate whether these chemical methods are effective or not? There is a dilution test that is used. So you have these metals or glasses or beads that are dipped in bacteria and dried. So you have the organisms on there. And then they are then placed in a particular disinfectant that we want to evaluate for 10 minutes at 20 degrees. And then they're transferred into a media. And so let's say these are the beads that we have. We dip them in bacteria and dry them, so they all have bacteria around them. And then they are then introduced into the disinfectant that we want to evaluate for 10 minutes at 20 degrees. And then after that, we introduce them into sterile media and then we see where the growth occurs. So that it's a good way of evaluating chemicals. Another way is to use a Dix dilution method. You will do this in lab, and that is where you have Dix that have been fortified with chemicals that are bactericidal or bacteriostatic. You place these on an agar that has the organism of interest on there. So let's say we want to test the efficacy of penicillin on, say, Staph aureus. You have to plate the Staph aureus on a plate, incubate it, let it grow, and then introduce your Dix that have infused with chemicals. And then you incubate, and then the region around the Dix of clearness, it gives you an idea how effective or not. But you have to measure this and compare it to a standardized table for these antibiotics. You have chemical methods, including phenols. The phenols are very effective against the membranes of bacteria, and they may be able to stay effective for a longer period of time. So the downside, however, is that they are corrosive and irritant and may cause mutation. So that's a red flag right there. Then you have the bisphenols. The bisphenols are derivatives of phenols, so they are derived from phenols. Example is hexachlorophene. And so they are used in surgical and hospital microbial control. They are more powerful. Gram-positive streptococci and staphylococci are all susceptible to them. You have triclosan, which is another antimicrobial soap that is used. We won't go too much into the antimicrobials right now. We'll tackle that in another lecture. Then alcohols. Alcohols are used in a lot of disinfectants, and they are effective against bacteria, fungi, and viruses. They are, however, not effective against fungi that form spores and bacteria that form spores. So when it comes to spores, you don't expect your alcohol-based disinfectant to be able to destroy them. And how they operate is that they destroy proteins and dissolve lipids. The most effective percentage in terms of the solution, alcohol solution, is 70%. So you start seeing effects beginning at 60% solution all the way to 95%. When you get to 100%, you don't see any effects. There is no effect, there is growth of bacteria. So this is our participation question for the week. 60 to 95% of alcohol in aqueous solution is an effective biocide. A biocide is a chemical or microorganism that renders a controlling effect on any harmful microorganism. So 60 to 95% of alcohol in aqueous solution is an effective biocide. But 100% alcohol is not effective in controlling microbial growth. Explain at least 2 reasons for this observation and include any references used in your research. Bacteria. Then you have some halogens, still the chemicals that are effective against bacteria and some spores. Iodine and chlorine are good examples. Iodine has been used for a very long time in treating wounds. Chlorine is a bleach which we use extensively, sodium hypochlorite, and it's also used to clean drinking water. Then we have some metals that are also used. So some elements, they're able to destroy proteins. Example is silver. Silver nitrate for a long time was used as an eye drop for babies to prevent blindness in case they picked some infection whilst being birthed naturally. But now we've moved on, and antibiotics are used more commonly, and mercury and copper are also used in some of these bacterial control methods. Then we have surfactants, and that's where the soaps and soap families come in. And what they do in terms of their um, um, mode of operation, how they work, is that they destabilize the surface tension, and so when they reduce the surface tension, it creates a very slippery environment for the microorganisms. There are some active agents in them, but mainly when the surface tension is reduced, it's very easy to wash most of these microorganisms off then. So soap is not an antiseptic, it is a degerming agent. And so it's the whole process of washing your hands is to remove these microbes, break the oil film on the skin, and help wash them off with lather. And up to about 20 minutes, you should be good. It's more important, especially in this time of COVID, that we know why we're washing our hands and how to do it properly. Then we have the quaternary ammonium compounds, which we call quats. So these are ammonium salts. They are bactericidal, and gram-positive bacteria are more susceptible than gram-negative, but they do not also destroy endospores. And we've had instances where Pseudomonas is able to grow even in these surfactants like quartz, having extremely no effects on them. And it's mainly because of the porins. You remember the porins that I mentioned earlier. You have aldehydes. They destroy the protein and damage the DNA or RNA. And representatives will be the formaldehyde, which is used in preserving biological samples and tissues. They tend to have a very strong odor, and that's very unpleasant to a lot of people. Those of you who have already done anatomy may have come into contact with formaldehyde already. Then there is glutaraldehyde, which is less irritating. It's more used now than formaldehyde. So it's used in disinfecting hospitals and hospital equipment when sterilization is not possible. Okay. And if they stay on the surface for 3 to 10 hours, they're able to kill spores. So that's a good thing. We have some that are oxidizers, and so they're able to destroy obligate anaerobes. Obligate anaerobes tend to infect wounds, and so they're able to kill them. Example will be the peroxide, hydrogen peroxide, and peracetic acids. Then we have some gases that are used as well. So you have ethylene oxide, which is used when you have heat-sensitive equipment that you cannot use heat to treat them. Then we'll use gaseous methods and gases such as ethylene oxide and chlorine oxide to destroy them. They're not as efficient as the liquid or the other physical methods that we use. However, they only use when it's very necessary, like an instance where you cannot use other methods to do it. And so there is low efficiency, and they can be toxic to those who are administering it. But they have to be administered in a very closed chamber for it to be effective and for protective reasons as well. So this brings us to the end of our lecture on microbial control.